All right, so let's get to the bigger paper, the more recent paper, that talks about if you have all these cross-reactive T cells, that means you have T cells that get through negative selection that could react with self. What's preventing them from doing that? And so there are differential roles of regulatory T cells. Regulatory T cells is the short answer. The longer answer is we have different kinds of regulatory T cells, and there's two kinds, including one that has to do with our favorite molecule of the year, which is the project we're going to be working on, which is the CURE project. So the thing about this is if we have peripheral tolerance, and if Tregs promote peripheral tolerance, if we can figure out how they work, then we can actually have some hope of solving immune diseases, autoimmune diseases in particular. This is called calming the storm. Here's 3 recent examples. There's an example of you have antigens that you give the person antigens that go to their liver, and at their liver, they're presented to Tregs at a greater rate. There, they go to the liver, they're presented to Tregs, and that makes the right kind of Tregs that you want to have to specifically regulate that particular antigen. Another thing you can do is you can make nanoparticles that have little MHCs on them. And the MHCs have little peptides on them that are the antigen that you want to induce the regulatory T cells. So you give those, and because they go through the periphery, I believe, they will activate T cells in the periphery. If it's the right kind of activation, you end up with regulatory T cells that come out of that. Notice that these are not going to the thymus. We're not talking about central tolerance, we're talking about peripheral tolerance. Then finally, if you have problems with your B cells, if you have lupus and you have overactive B cells making overactive antibodies that are attacking your own Fc regions of IgGs, then why don't you find a way to destroy those B cells? Now there's an approach, we're going to talk about this in the cancer chapter that's going to be on Monday. But there's an approach called chimeric antigen receptor T cell therapies that can target cancer. Well, why can't we use that to target B cells? In fact, some B cells are the ones that we want to attack. And so if we can target cancer, why can't we target B cells as well? And we can use this thing where we target the T cells. Exactly how we do that is more of a topic for the cancer lecture coming up. But and there's another way, so like Mark Davis is working on how do these peripheral tolerance mechanisms work? And in the Standing on the Shoulders of Mice, he asked the question, why do we have these? Now we start with evolution. There's a famous quote, nothing in biology makes sense except in the light of evolution. Evolution can help us understand why we have some problems that we have. So for example, if you think about things from that level, autoimmunity, we have all these T cells that are in the periphery that have to be turned down because they're autoimmune. They react with self. We constantly have to be turning them down. Why do we have them? Well, maybe it's because why do we have them at all? Well, autoimmunity is not a problem for children usually. It's usually a problem for adults. And TCR, autoimmunity could not be a driver of what TCRs you have. And Tregs have TCRs. So we're thinking about evolution doesn't care if you develop autoimmunity because it cares that you live long enough to get to the point where you're passing on your genes. And autoimmunity is not a children's problem. So what is a children's problem is infectious diseases. And this is also a children's problem that has been addressed, that has gone far down for our continent at the very least. Because we now have vaccination, antibiotics, sanitation, children who used to die before age of 5 would live. In other words, infectious diseases used to be a much bigger problem. We probably evolved to have a more active immune system against infectious diseases. So if there's a way for our immune system to care more about infectious diseases and less about autoimmune diseases, it would make sense for it to evolve that way. And it turns out that if you think about it, peripheral immunity may be you have better infectious disease reaction if you have more peripheral T cells that are messier, that they react with as many things as they can. Because if you have a self-reactive T cell that you're keeping in check by keeping it around, but keeping it turned down, then someday you get infected by a novel pathogen you've never seen before. If you have more of these self-reactive T cells, there's a chance that one of these self-reactive T cells will accidentally be able to react with the pathogen, and you'll be automatically already protected from it without having to adapt your immune system to it. In other words, a broader cross-reactivity at the TCR level is actually better if you're more worried about infectious disease than you are about autoimmunity. Once you have the broader immunity, then you have you want to tone it down. Now we've made the broader immunity not as important because we've eliminated the infectious diseases in other ways. Now we have the problem of our immune system that was before it was active enough. We've turned down the problems it has to do. So our immune system is now too active and it starts to attack ourselves. I want you to see this is a key concept. And it's how peripheral immunity works, or peripheral tolerance works. So Davis argues that basically, part of his argument that human peripheral tolerance is important, but we haven't studied it because it's hard to study. And he's saying that it's more important than mouse central tolerance, yet we know more about mouse central tolerance because we can operate on the thymus, we can operate on mice, we can make inbred mice that have defined MHCs rather than humans that have undefined MHC compositions and are walking through we're wading through a whole bunch of pathogens every day. So negative selection is real. It's important for central tolerance. It's important in both mice and humans. But its effect on humans is exaggerated. Cross-reactivity means that not only do you have some T cells against diseases you've never seen, but you also have some T cells against your own proteins. It's a double-edged sword. And so because your immune system is not perfectionist, it will tolerate some autoimmunity. It's evolved to let some self-reactive T cells through because it's better to react against an infectious disease than to react also against a self-antigen later. It's better because at least you live to react against the self-antigen, right? So instead of eliminating all autoimmunity, what the system does, it doesn't turn up negative selection. Instead, it turns up the sort of regulatory periphery negative selection, equivalent of negative selection. So instead of eliminating all autoimmunity, the system edits down the peripheral T cells. That leads to the question of how does it turn down the T cells once they get out of the thymus into the periphery? How does that work? We know it's Tregs, but are there different kinds of Tregs? It turns out that Mark Davis has an answer to that just came out. It was only accepted in 2024, and it was actually published technically this year. So now we have it. You're probably the first people to hear about it. So this is about two Tregs that work differently in sort of opposite ways. Each of them has a characteristic gene. They're CD4 positive Tregs, so they're helper Tregs, and they have FOXP3 as their main gene. If you turn off FOXP3, the CD4 positive Tregs also turn off. CD8 positive Tregs, they have KIR receptors, so they're CD8 positive, KIR positive. They also depend on granzyme B, which is the GZMB gene. Sometimes the name sort of makes sense. If the GZMB gene is turned off, these CD8 positive Tregs, these are also turned off. The good news is we now have a technique called CRISPR. It's a molecular biology technique, so you can learn about it in those kind of classes. For us, it's basically a delete button for particular genes. We can program CRISPR to go after and delete, knock out a particular gene. And so that's what they did. They deleted FOXP3, and they looked at an immune system without CD4 positive Tregs. And then they deleted GZMB, and they looked at the immune system without CD8 positive Tregs. And they found they could make sense of the differences. The other thing is that what are they using? And of course, after Mark Davis wrote a whole thing about how mice studies are overblown, you might expect that he didn't do that many mouse studies in this. He actually worked with the tonsil organoids. Remember that he actually used that in the previous paper we talked about. These seem to be very versatile because they allowed Mark Davis and colleagues to be able to separate out what are the effects of CD4 positive Tregs versus CD8 positive. And these are their primary conclusions. I'm going to give them to you up front, and then I'm going to show you the data that supports this. So when they removed the CD4 positive Tregs, they saw that you got a bunch of autoantibodies, and not more did you just get more autoantibodies, they also had higher affinities. And this is a clue to how the immune system works. And then you also got severe inflammation. If you're getting rid of the Tregs that calm down an inflammation, then no surprise, you're going to get severe inflammation if you completely remove them. Now, when you turn to the CD8 positives and you deleted the gene GZMB, you also end up with severe inflammation. And that's not really a surprise. What's interesting is how things are different in that case. They also saw that you saw a large increase in a particular kind of helper T cells in follicles. Okay, so that's their primary finding of what goes on. And they're saying that actually from this, it looks like the CD8 positive Tregs are the ones that are really in control here. And also it makes sense. The ones that have the power to kill other cells are the ones that have the power to control the immune system. There's also one more thing, and I'm just going to mention this once. They also saw a rise in plasmablasts, but they argue this is an indirect effect, and this is getting too complicated for us. So we're not going to talk about that. But the final thing that they did see that we'll talk about, they saw both effects were larger in women's tonsil organoids than men. This goes right along with what we said. Autoimmunity usually is a bigger problem in women than in men, and that would go right along with autoimmunity is mediated by these Treg cells. Okay, so here's a figure. Let's go through the figures that are key to showing the data, and you can look at how strong this data is. I want you to look at these. So let's look at figure 1A in the upper left corner. This one, they're just looking at how many CD4 and CD8 positive Tregs do we have. And there's more. They looked at PBMCs, remember those are blood, versus tonsil organoids, and they found out actually these are probably just regular tonsils. We haven't gotten to the organoids yet. They found that the CD4 positive Tregs, they like to hang out in the tonsils, but they also found out that, see the next one over is the CD25 negative. There's still a lot of T cells, there's almost 50% that don't have CD25 negative. And this is one of the things that they talk about that makes immunology complex. Previous studies have said that these Tregs need to have CD25. Well, those previous studies weren't entirely right, so we shouldn't really look at CD25 to see if we're looking for these particular kind of Tregs. Okay, but we found the one kind of Treg, CD4 positive Tregs, and we found it both with and without CD25 in both tonsils and PBMCs. It likes to hang out in the tonsils. Then we looked in the T follicles, and we looked for, we look for these T follicular regulatory cells. And these are the kind of cells that we see as increased when we get rid of a certain kind of regulation. And it turns out that those kind of T cells, whatever they are, what matters for us is they like to hang out in the tonsils. So if we're looking for them with tonsil organoids, it's reasonable that we'll see an effect with these. If we're looking for them in the blood, they don't even show up in the blood. Look at B, and the blue dots are way down for PBMCs. So there's more of these more of these TFRs in the tonsils. And so then we move on to the CD8s. Those are all about CD4s. The CD8s down there are the CD8 Treg cells. They're in low percentages, but they can be found. It's not zero in both the PBMCs and the tonsils. And also the granzyme B positive T cells. The blood is actually going to be expressing more of the granzyme B positive in the tonsils. They just aren't using it very much at that time. So that just sets up, this means that they found them. They found a lot of them in the tonsils. They found enough of them in the tonsils at least to move on. And this is the kind of data. This is the first really key. This is the inflammatory. This shows that when you knock out these genes to knock out either the CD4 or the CD8 positive kinds of these Tregs, you will end up with inflammation. And so what you see is the wild type cells with all their genes. Then you see the red cells, the red bars show that you've knocked out the FOXP3 gene. And you see the CD4 positive T cells, they're going to be more inflammatory. Notice this is, I believe this is not Tregs, this is just regular T cells. This is just looking at different kinds of T cells. And the main thing to notice for both the blue FOXP3 knocking out and the, or I said it backwards, the blue GZMB knocking out and the red FOXP3 knocking out, those numbers are higher. You have more T cells that are expressing the inflammatory phenotype. They have these receptors only when they're inflamed. And so you see that all three of these kinds of cells, they react. They don't like it. They are turned up, and they might like it because they probably like to be activated. But when you knock out both of these genes and knock out both of those Tregs, the Tregs are holding back the other T cells from becoming too inflamed. Inflammation is up on this graph, and you can see that there's an increase in all these kind of T cells, including these special follicular helper T cells. So just look at that. There's the fold change in the CD4 positive T cells. The T helpers are more, they're showing more inflammatory markers. The CD8 positive T cells are looking more inflamed as well. And then in the middle, you have the T follicular T cells for both blue and red, both genes knocked out. We end up with more inflammation. This is cool. But now we want to say, so we saw severe inflammation in both cases. But now we want to say, how are the CD4 positive Tregs different? Why are they associated? What are they associated with? And they found out that when you knock out FOXP3, you end up with autoantibodies all over the place. This is where they show this. So this is where you have, you're comparing female and male. There's a lot going on. So the orange bar is where you do nothing to the cells and you're just seeing how many autoreactive B cells you have. If you look at the blue bars is where you have autoreactive B cells after you've activated them with an influenza vaccine. Basically, you take a influenza vaccine that's really the live virus, but just attenuated, and you're giving them something to fight against. And you see in these cases that the blue bars are still down. You don't have more autoreactive B cells. The B cells are all reacting to the flu. Okay. But then you have something where you give them the flu vaccine and you give them autoantigens to see if they react against those. And the red bars are where you have the autoantigens, and for the GZMB, everything's still down low. But for the FOXP3, all of a sudden, you have a higher number of dots. And you see that the when you have the FOXP3 and you stimulate them with an influenza vaccine virus, and then you add autoantigens, that these B cells are going to be reacting more to autoantigens when you provoke them with the virus. That bar is up. And notice also they break it down by female and male. They have at least two. It looks like there's only two. Yeah, there's only two. This is only 3 or 5 examples. There's 3 males and 2 females. So you can't say anything from this alone. But it sure looks like those 2 red dots above the red bar are higher than the 3 black dots in the same red bar. Everyone's reacting more. Everyone's producing antibodies to the autoantigens when you knock out the CD4 positive Tregs. But it sure looks like the women have it worse. So if all these antibodies are being made, let's take those antibodies and let's look at them. And so they took the they took the cases where you have more antibodies, and these are antibodies against self, autoantibodies. You're taking them from the wild type tonsils, the ones with the GZMB knocked out and the ones with the FOXP3 knocked out. And look at this. Wild type antibodies, these are the autoantibodies that you've induced from wild type, and they bind. And by the way, the higher up on the graph on the left, it just shows antibody binding. The higher up the graph, the tighter the antibodies are binding. More and tighter. And it turns out that, yeah, it's not just more, it's also tighter. You can see that the antibodies from wild type mice bind with the red curve, which is lowest. Then the autoantibodies that are made in the GZMB, and I said mice, my goodness, I'm so used to mice, I mean tonsils. The GZMB tonsils, where they have the CD8 knocked out, they make autoantibodies, but they're binding more, they're higher than the red line, but the green line is the highest, and those are the ones from the FOXP3 antibodies. And you can see that it's definitely the highest. Those are definitely higher affinities. This is really interesting. And what I love about this is they also looked at the half-lives. Remember that we talked about half-lives with MHC peptide? I talked about in grad school, I would bind the peptide to the MHC and I would wait for it to fall off. The faster it fell off, the weaker the interaction was. Well, they also looked at the half-lives for these different kinds of antibodies. They bound the antibody to an autoantigen and they let it fall off. They saw how fast it fell off. The wild type ones fell off really fast. The GZMB ones fell off medium fast. But the green ones, the FOXP3 ones, they took forever to fall off. This is why they said that removing the CD4 Tregs, they had something to do. The Tregs, when you take them away, you end up with more autoreactive antibodies and worse autoreactive antibodies because they bind more. I just really like this analogy. Just they're measuring the binding affinity of antibodies. They're actually quantifying how tightly they bind. And this is my wheelhouse. I like to quantify the things. This is what I do. I like to quantify immune reactions. And this is definitely a clear quantification. And it shows you a clear difference between the green and blue. The green is where you have CD4s knocked out. The blue is where you have CD8s, both regulatory. Oh, and by the way, this is because this is a flu vaccine that they're looking at. They're actually looking at the antibodies. Oh, and I might have said this is related to the fact that the autoantibodies are higher, but all antibodies are higher. Technically, what they're measuring here is they're measuring the other centered antibodies, the ones that are binding HA, peptides, and epitopes. So I said that slightly wrong, but the whole point is that all antibodies, auto and regular, are going to be not just increased, but increased in affinity. They're more potent. And this is interesting because it means to say that you don't want the most tightly binding antibodies. You're not trying to win a competition for the tightest binding antibody. In fact, you want something that will bind, but it'll eventually let go. Because if it binds and never lets go to an antigen protein, it might bind and never let go to a self protein. The idea of self-reactivity may limit the antibodies that the tightness of antibodies that is useful for us. And notice that what's happening here is they knock out part of the Tregs and they end up with self-specific antibodies. Isn't that just like the case study for the AIR gene that we had, where the child Robert Jordan was a pseudonym that was AIR deficient, had self-specific antibodies. You're seeing more antibodies and they're tighter binding. And it looks like you're seeing the exact same thing going on here. In other words, what AIR does for negative selection might be the same thing that GZMB does for peripheral tolerance. Very interesting. And just remember, it's not about the highest affinity antibody, because the highest affinity antibody is more likely to have high affinity accidentally to something that's almost like the antigen, but not quite, some self-antigen. And so if you have a too tight binding antibody, it's more likely to be an autoantibody under certain circumstances. I think that's really interesting. And for biochemists, by the way, we know where this number is, but I haven't discussed what these numbers mean, so I'm not going to focus on them. Basically, if your immune system makes too many antibodies that are too tight to pathogens, then you're more likely for one of them to accidentally bind too tightly to a self-antigen. And that, again, is because we only have 20 amino acids, only so many surfaces are presented to antibodies. there's going to be some overlap, there's going to be some mess inside the human body. So instead of tamping down on it, we have Tregs that will keep it from happening. It looks like the CD4 positive Tregs are there to keep it from happening in the periphery so that we don't get too many antibodies against our own organs. The last thing to talk about is how the women have more autoimmune diseases than men do. And so they tested this in their own little tonsillar system. Their little tonsils, so they had some from women and some from men. And they tested how their frequency of autoreactive B cells to particular autoantigens. You notice that the one in the middle is histones, which is something that DNA wraps around, should be inside the cell. You should not have autoantibodies against histones. It doesn't do you any good. And in fact, it causes you problems. If histones get exposed, you react with your histones. So they looked at the frequency of these in their tonsil systems, and they found out, look at that middle graph. The red dots, the female tonsils, reacted far more. In fact, there's only one male dot that's even in the range of all the female dots. The females had more B cells that would react to histones. And for these other autoantigens on the side, it's not as stark of a difference, but it's definitely higher in general, that you have more red dots that are high, meaning more women that will actually have B cells that react with their own proteins. They showed this with a slope and they wanted to show this, that this actually also develops over age. Remember, we're talking about autoimmunity as being a problem with an older immune system. If this is right, then older people should have more autoimmunity, and we know that they looked at this specifically for, do our tonsil cells produce autoantibodies that are from our tonsil cells from the older people produce more autoantibodies as well as older women. And so they made little slopes where they have the age of the person who gave the tonsils next to the on the x-axis, and they have red women versus blue men. And as you're going up, you're detecting the antibodies by putting the antigens in the ELISA plate. They're literally doing this technique. I told you to watch out for it because they'd be doing it. So this is an ELISA plate. They put the antigens on the bottom of the plate, and they measured how many antibodies the tonsils would produce that would bind those antigens. And there's a lot of mess to this, but I think it's still a clear result. The red line for women is always above the blue line for men, and it's also going up with age, always, for these 4 very different autoantigens. This implies a general effect, like the AIR gene has a general effect for negative selection. The FOXP3 gene, in this case, has a similar effect when it knocks out the CD4 positive Tregs. And here's I want you to be used, that's why I'm quoting the paper, I want you to be used to the way they use language, and I want to try to explain it to you. They say directly, our results highlight the differential roles of CD4 positive and CD8 positive Treg cells in regulating autoimmunity. The GZMB knockout, remember that is the one that knocks out just CD8 positive, and it shows that the CD8 positive Treg cells are the major factor controlling T cell tolerance, especially for autoreactive CD4 positive T cells. CD8 positive Tregs turn down autoreactive CD4 positive T cells. By the way, that was one of the charts that showed the CD4 positive. I believe it's figure 2. So their previous work, and this goes with what other people have seen in other diseases, so SARS-CoV-2 and influenza, in both those cases, they've seen more CD8 positive Tregs. That means CD8 positive Tregs are part of your immune system keeping inflammation in check, and doing it by keeping autoreactive CD4 positive T cells from freaking out too much. So they're involved with infection and autoimmunity. And remember how this goes along with the idea that infection often triggers autoimmunity. If this is the case, this could be CD8 positive and CD4 positive Tregs might be part of the reason why infection will do this. And in fact, we can look at it as the first trigger would be when infection disrupts the CD8 positive, KIR positive T cell activity. Remember, in infection, you want that regulation to go away. So infection might disrupt that. They're holding back the self-reactive CD4 positive T cells. When the holding back CD8 positive, KIR positive ones go away, then the CD4 positive self-reactive ones get leash because you're unleashing the whole immune system. Not just the ones that react to the infection, but the ones that react to self as well. This gives us hope. Maybe we can target this because as people get older, they have more of a problem. We don't want this to happen. They're not getting infected. We don't want them to attack themselves. Maybe we can use this by targeting these cells, and maybe targeting KIR is one of the possibilities. They actually say that in one of their papers. And we are going to be looking at KIRs. So even though this is complex, it's actually going to tie in together. All of immunity is complex, but it ties together. And that's the double-edged sword of immunity, because it's really cool, but it's also really complex. So I hope you have, if you need to listen to this again, read it, use your eyes and ears, study it. We will definitely have some questions about this on the final, and I look forward to coming up with them so I can attest that you see the complexities of what's going on. By the way, autoantibodies are probably associated with CD4 positive Tregs, and those often show up before autoimmunity becomes really bad. So autoantibodies produced by CD4 positive Tregs can be sort of the non-trigger event, like the low-level thing that says you might have a problem with this someday, but they don't get out of control until the CD8 positive, KIR positive T cells are the ones that actually trigger the autoimmune thing by holding back the well, they're actually, what they do is they hold back. So when you hold them back, they hold back the holders back. It's, believe me. Let me try to say that better. CD8 positive, KIR positive T cell activity normally is keeping inflammation from going out of control. But if you have an infection, you want your inflammation to be less in control because you want your immune system to be activated. When you have autoimmunity, you have the same thing happening. You don't want it to happen. If we can control it, then we can possibly control autoimmunity. See why I said the immunology is back and forth? But if you get this, then you get the whole thing. So I'm going to have a key idea question about this and read their own words. Read the words and listen to me try to explain it. And yeah, have a good weekend.